students, welcome to lecture 5. So in the previous video we started with the differential scanning calorimeter. So this is nothing but your thermoanalytical technique, thermoanalytical technique, okay, which is in which the difference of difference in the amount of heat required. So we have studied that it's a difference of amount of heat required, okay, amount of heat required to increase, amount of heat required to increase the temperature of sample, increase the temperature of sample, okay, temperature of the sample and the reference. So this is how we have done this in DSC as a function of temperature, as a function of temperature. So this is what we have done, and both the sample and the sample and the reference here are maintained nearly the same temperature. Okay, so we have maintained nearly the same temperature throughout the experiments. Okay, so this study, this technique is to study what happens usually for the polymers, okay, to the polymers or sample upon the heatings. So this is used to study the even the thermal transition, thermal transitions, so the study of the thermal transition in polymers. Okay, so this what in the previous video we have seen. And the for example, if you see the melting of the crystalline polymers, okay, we are using glass transition temperature and in the crystallization. So this is what in the previous video we have learned, and the result of this DSC, differential scanning experiment, is a curve of heat flux. We have learned that is a curve of heat flux versus temperature or time. So this is what, okay, so heat flux versus temperature or time. And if we have seen that there are two different conventions, that is your exothermic reaction, where the sample show the positive or negative, okay, peaks, okay, depending on the kind of technology, depending on the kind of technology we use. Okay, so this is what, either it show positive exo or negative. There are two peaks we have seen here. Okay, it depends on the technology whatever we are using. So we are seeing that the exothermic reaction, what is happening in the sample, that is release of the heat. So it may have the positive or negative peak. So this is what, okay, so this curve can be used to calculate the enthalpy of whatever the curve we are going to get. So it is helpful to calculate the enthalpy of transitions. This also we have learned, enthalpy of transition. This is very much important, is it right? And which is done by integrating the peak, so corresponding to the given temperature. So here the enthalpy of transition can be expressed as we have seen, that is your delta H is equal to Ka. So this is what we have learnt in the previous video. What does this delta H mean? Heat of enthalpy, okay, of transition, and K is calorimetric constant, and this calorimetric constant varies. It varies instrument to instrument, and it is determined, okay, by analyzing the and by analyzing, it is determined by analyzing the, okay, the well characterized material, well characterized material, well characterized material of known enthalpy of transition delta H. So this is how we take it as a constant, calorimetric constant here. And A is nothing but area under the peak. Okay, so A is nothing but area under the peak, and we have seen in the last video that is directly proportional to heat absorbed or evolved in the reaction. And the height of the peak is directly proportional to rate of reaction, which is help in the study of chemical kinetics here. And this is what in the previous video we have learned. And the heat flow is that we have seen that the formula, that is Q, heat flow supplied per unit time. Okay, so that is what, this is called heat flow. Okay, so the heat flow heat flow in the sample, that is given by Q, supplied per unit time. And whereas heating rate, okay, so this heating rate, you can see that is this which is given by the temperature delta T per time, change in the temperature delta T per time, that is heat flow is given by Q by T, and heat flow delta T, that is Q by T, and heating rate, that is delta T by T. Okay, delta T by T. Now if we divide these two here, so in the yesterday's class we have done it, so when we divide these two, that will give you Cp. Cp means what here? That is heat capacity, and you know that is when we divide this Q by T divided by delta T by T, T get cancelled, and you are going to get you are going to get this Q by delta T, and this is nothing but your Cp. So when you do this, so that is your that is what your heat capacity. Okay, so this is how we have done in the previous video. So this is what about the DSC, and today if we go for the closing of this DSC, the final part here, so we can go for some advantage. Okay, so advantage, so you can take it here, that it is more advantage of this, it is more popular technique, it is more popular technique. Okay, so it is more popular technique because it's a speed, because of its speed and simplicity and availability and availability. Okay, so it is more popular technique than the DTA or TGA. So DSC is more recognized because of the speed of its technique, as well as simplicity and the availability. The second is a different kind of sample, as solid, liquid, powdered, thin films. Okay, powder or thin films. So this can be tested in variety of environment, of environment. Okay, so like air, vacuum. Okay, like air, vacuum, or inert gas, etc. So that's why this is one advantage, that that the solid or liquid, powder, anything, or thin films can be tested in various variety of the environmental, like air, vacuum. So this also becomes a very important advantage of this, and a wide range of temperature can be used. Wide range of temperature can be used. Okay, wide range of the temperature can be used to analyze phase transition, to analyze phase to analyze the phase transition, that is your glass transition temperature, TG, or crystallization temperature. So this is what we can see, the advantage, okay, of this DSC. And if you see the disadvantage, so not more, so disadvantage, but only thing is what you can say, the accuracy and precision has somewhat the negative impact on this DSC. Okay, since the metal and everything, the sample is taken in the way the sample is taken in the container, so we can say that the proper accuracy and precision getting results is a little bit problem here. And even the conditions, lab conditions, and humidity, cooling process. Okay, lab condition, if humidity present, then cooling process, change in some cooling process, this all may lead in what you say, on the accuracy, precision not exactly giving the results, and that is one of the disadvantage of the DSC. But it is not, it is very rare, very less, so we can negligate this disadvantage over the advantage. So advantage is far here. In most of the industries and even in the laboratory research, we can see that DSC is used maximally rather than TGA or the DTA. So this is what, and if you see the application of DSC, now this is used in most of the in the characterizing materials. Okay, so this is what, like in food science. Okay, in food science or pharmaceutical industries, research lab, paint industry, and like this. In most of this, we can see that DSC is used to see the change of phase transition of the substances. Okay, so this is one advantage or the application I can say. And the second is, as I said now, that is your determination of phase transition, determination of phase transition. That is, as I said now, glass transition temperature or crystallization or melting, these all things. That is your physical and chemical properties change can be easily determined with the help of DSC. And even as I said, that is, it is helpful for determination of reaction kinetics, determination of reaction kinetics. Okay, so determination of reaction kinetics like exothermic, endothermic nature, and some etc we can put. So these are some of the applications of DSCs. Okay, hope you understood in this this conclusion part of the DSC. Now I think we have completed with good examples the three thermoanalytical thermoanalytical concepts like TGA, DTA, and DSC. Okay, so if any problem or in net problems, if CSR problems is there, we will solve definitely in some other in future videos depending on this, and most of the questions will be definitely asked on this in the net examination. Okay, so we will net target examination. So we will solve this also in the future videos. Now next we'll go for voltammetry. Next concept of your syllabus, that is voltammetry. Okay, so this is also electroanalytical technique. Voltammetry is nothing but electroanalytical technique. Okay, so fundamentals of voltammetry he has given in syllabus, so that is what we will learn. We will start with this basic concept here. So this voltammetry, it is a information regarding an analyte. It gives the information regarding the analyte. Okay, regarding the analyte. So this is determined by measuring the current. This is determined by measuring here. This is important. Voltammetry means the measuring the current. Okay, analyte by measuring the current with respect to variation in potential, with respect to variation in potential. This is very much important. Okay, so there is the change in the current of by the analyte with respect to the variation in the potential, and that study we call it as voltammetry. And the data represented in the form of voltagram. Okay, whatever the data we are going to get, it is measured in the form of voltammogram, and at a plot, voltammogram, and that is the plot what we put for this for this voltammetry is between the current produced by analyte, that is the current produced by the analyte, and the potential of working electrode, and the potential of working electrode. Okay, this is very much important. So what plot we are plotting here is your the between the current produced by the analyte, what we are taking in the electrolyte, versus the potential of the working electrode. So this is what the plot we take in the voltammetry. Now this working electrode, reference electrode, and auxiliary electrode is used here. Working electrode along with reference electrode and auxiliary electrode, that is your 3 electrode, 3E. Okay, so this is what we are going to use in this voltammetric process. So if you see in this way, so you take this here one, this is one electrode, this is one electrode, and this you can take here A. Okay, and this is connected to battery, and you can see this is connected here, and this you can see this is connected to voltmeter, and like this, and you can see, so this is your working electrode. I will write this WE. This is your working electrode, and this is your reference electrode. This is your reference electrode, and this is your auxiliary or counter electrode. We call it as a counter electrode, or also it is called as auxiliary electrode. So this counter electrode, so it maintains the constant current here, and see here it is maintaining all the current between this reference as well as the auxiliary as well the reference electrode here, and whatever the change is happening in this working electrode which is going to with the analyte here, change of the analyte here is been detected or is been taken care by this reference electrode with reference WE. So we can get the plot here, we can get the result here. So this is what about, and you can see that whatever this counter electrode it is made up of any content here that should not have any reaction with the analyte solution and conducts, and it should conduct the current well. This is very much important. You can see here whole thing is maintained by this counter electrode here. So you take any of this material of this you can make it. Only thing is what, this auxiliary electrode should not react with the analyte, and that is what, and it should conduct the current very well. So this is what about the auxiliary electrode or the working or the counter electrode here. Okay, so when this working electrode is made up of DME. What is this DME? DME is dropping mercury electrode. If you convert this working electrode into dropping, as you take it, okay, as a DME, the dropping mercury electrode, so we call that voltammetry is called polarography. Polarography, as in second semester I think you have learned this polarography in learning the stability of the metal complexes. So that is what, where this voltammetry, where this working electrode, if you take it as a dropping mercury electrode, so that process we call it as that voltammetry method is called as a polarography, this way. Okay, so this is how we can see the basic of the voltammetry starts in this way. And if you see the principle of electroanalytic chemistry is the direct, if this is a principle we can give, that it is a direct conversion of chemical information, direct conversion of chemical information, direct conversion of chemical information into electrical signal, into electrical signal. Okay, so this is what important here. And analyte is dissolved in electrolyte solution. Whatever the analyte we are concerned is dissolved in electrolyte solution, solution. Okay, this is what dissolved to get the results of the changes of the analyte, to study the analyte here, and the detect the undergoing oxidation reaction. So whenever the analyte is been varied with the potential, then the whatever the changes takes place like oxidation or reduction, so that can be detected here. And when the oxidation occurs, it is called anodic current. Okay, when oxidation occurs, it is called as anodic current, and cathodic current if it is cathode or if it is reduction. And the difference potentiometry, that is difference potentiometry, that difference in electrode potential is measured here. Whatever the potential is, the difference will be there, it is been measured. And the voltmeter, that is cells current, regarded as a actually alternating cells potential. So this is not we will not include here. And this voltammetry, that is cells current voltammetry, was first coined in 1940 by H.A. Leitinen. Okay, so he was the first person to coin this voltammetry in 1940. And the plot, as I said, so it is nothing but potential versus time, that is linear plot sweep. So we are going to get in this way. Okay, and we have seen the one more thing is what, three electrode setup, like in there. Now just we learned that is this one, three electrode setup. This is what, three electrode setup we call it here. And this voltammetry experiment we investigate by half cell reactivity of the analyte. Half cell reactivity, investigate the half cell reactivity of analyte. Okay, so we will give that half cell reaction. We will learn this whenever we go for the clear example here. And it's a study of current versus function of applied potential. Okay, so this is what, and the shape of the curve depends on the speed of the potential variation. So the shape of the curve also, whatever the curve we are going to get during this, so we'll see that. Okay, so when we do take an example and do, so sometimes we get the curve like this also. Whatever the curve we are going to get during this experiment, it depends on the speed of the potential variation. Now if you see the types of voltammetry, okay, types of voltammetry, among which two voltammetry is there in your syllabus, that cyclic voltammetry and stripping voltammetry. But if you see the types of voltammetry, very different types of voltammetry we can see, that is some of them are linear sweep voltammetry. I will give one by one. Linear sweep voltammetry. Okay, then there is a staircase voltammetry, staircase voltammetry. Then there is square wave voltammetry, square wave voltammetry. Okay, then there is cyclic voltammetry, then there is anodic stripping voltammetry, anodic stripping voltammetry. Okay, then there is cathodic stripping voltammetry, cathodic stripping voltammetry. Then there is adsorptive stripping voltammetry, adsorptive stripping voltammetry. Then there is alternating current voltammetry, alternating current voltammetry. Okay, then there is polarography, polarography. Okay, so even there is rotated electrode voltammetry, rotated electrode voltammetry. Okay, normal pulse voltammetry, normal pulse voltammetry. So even differential pulse voltammetry, etc. Differential pulse voltammetry, like this. So these are different types of voltammetry, among which for your syllabus we can see they have taken as a cyclic voltammetry you are going to learn, and stripping voltammetry you are going to learn in this voltammetry, among these different types of voltammetry. But you should know the different types of this voltammetry. Now if you go for the application of this voltammetry, so you can see nowadays the voltammetric sensors. Okay, so voltammetric meter sensors is used in the industries and research. You can see nowadays in industry and research the sensors, voltammetric sensors for detection of specific you can say analysis of various organic and inorganic analytes, for determining of various organic and inorganic analytes. Okay, so this is how you can see the voltammetric sensors is being used, and even the oxygen electrodes. Okay, in determination of dissolved oxygen, in determination of dissolved oxygen in various aqueous environment, various aqueous environment, as you can see in sea water. Okay, and even water, even blood. Okay, even sewage effluent from chemical plants, sewage effluent from chemical plants maybe, or sometimes effluent from soils, and even the biomedical industries, environmental biomedical industries. Okay, then environment research and clinical medicine, these all things we can see in all these things we can see there is application of voltammetry. Okay, so this is how I think now more than sufficient for the introductory part of this voltammeter. Okay, so and principle fundamental of the voltammetry. In the next video we will learn the main part of your voltammetry, that is your cyclic voltammetry. Okay, so this is what we will see in the next video. Okay, thank you.